Hello everyone, and I hope that you're doing well. Today we're doing a CSEC biology paper. It is a paper 2 from May, June 2019. Now please, before you start the examination, you should always remember to read through the instructions carefully. And if you should have any questions or queries, then you should clarify those with the examiner before the examination begins. Now let us jump to section A, question number 1. And for this question, he said figure 1 shows apparatus that was set up to measure the rate of transpiration of a potted plant located in a room beside an open window over 12 hours period. Now, if you observe the diagram carefully, realize that this is a well-watered plant. The pot with the soil is also tied tightly within a sealed polythene bag. And you're going to use here a beam balance. And of course, once you're going to use a beam balance, then what you're checking for is the difference or the change in mass. All right. And so part 1 asks us to describe the procedure that uses the apparatus shown in figure 1 to investigate the rate of transpiration of the potted plant within the 12-hour period. And a point to note, if you go to measure or check the rate of transpiration, this is a mass method, the change in mass method as mentioned before. And so anything that you're going to do here with the beam balance must include mass. All right, and so the first thing you're going to do before you start the experiment is to ensure that you balance the beam balance. And to do that without any form of weight, which is the plant in this case that is placed on the beam balance, you're going to move all the counter masses to the 0 marks. After you have done that, you're going to turn the 0 or the tier adjustment knob until the pointer aligns with the 0 mark, which we call the balance mark. After that, you're going to place the plant on the beam balance and record the initial mass. And after that, you're going to place the plant in the designated area, which is the window in this case. And then you're going to measure the mass of the plant after a specific time elapse or a specific time period. And then, of course, you're going to repeat step 5 for a specific number of times. And this will be over 12 hours period. So if you're going to measure every hour, every half an hour, whichever it is, but you have to measure in a specific time interval over that 12 hours period. And after that, you're going to record, during the experiment, you're going to record the masses and determine the rate of transpiration after that. And a point to note right here, you're going to use a suitable formula, such as the one given here. Rate of transpiration equals to the change in mass over time. All right, and you can say average change in mass over time. All right. Now, that's just a 4 marks question. Again, just outline exactly what you're going to do, but it's always good to talk about zeroing your apparatus or make sure your apparatus is clean. Things like that are very important. Now, for part 2, is a state 1 precaution that should be taken to ensure that the results are accurate. And one is to ensure that the bag is tightly sealed. That's very important because if it's not tightly sealed, then definitely evaporation can take place from the soil itself, and that is not necessarily coming from the leaves or from the shoot of a plant. So hence that will lead to error. Of course, you're going to use fresh and healthy plant as well, because if a plant is not fresh and healthy, for example, if it's wilted, then there's no way you're going to have further evaporation or transpiration. And you need to record the masses at the same time intervals. You can't measure 1 half an hour and then measure 1 hour and 2 hours. No, same time interval. Very important to do those things. All right, and the next thing here, part 3, is it suggests 2 factors that may affect the rate of transpiration of the potted plant. And so you could have temperature, humidity, wind speed, and light intensity. All right, and so any of those 2 will be correct. All right, notice the plant was well watered, so I didn't put water or available water because the plant was well watered. All right. Now for part B, it said the graph in figure 2 was constructed using the results obtained from the investigation in A, which measured the rate of transpiration over a 12-hour period. And so the graph that is given here, if you notice this shape of the graph, you have pretty much a decline going down, and then it kind of stabilizes, and then a slight decrease right here as well. And then it kind of stabilizes again. All right, and this is the time on the X and the weight of the potted plant on the Y in grams. So you want to make sure you know that. The units that are used are what they're actually measuring. And so to go in the question, Here now it said draw a suitable table and record the results presented in the graph in figure 2. And so from the graph, I obtained these values. Again, you can always go back and observe the graph. And so what I did, just want to point out a few things here. You can draw a table anyhow once it represents the information properly and accurately. And so I have time of day in hours here, and I separate my AM from my PM and also my noon, if you realize that. And then I have a weight with the weight of plant here and the weight during the AM period, the weight during at noon, and the weight during the PM period. And so again, it's up to you how you want to do your graph. You can do it in any way as long as it is showing the accurate time in relation to the weight or the change of weight. All right. So again, just be creative and be simple as possible and straightforward. Now, during what time period was the rate of transpiration the highest? And if you go back to your graph again, you're going to realize that that is between 9 a.m. to 11 a.m. And the slope of the graph or the steepness of the graph will indicate that here. All right. There's a big jump. Matter of fact, even the table shows that in terms of the numbers. If you look at the numbers carefully, you will see those between 9 and 11. So notice a 20, difference by 20, difference by 20, and there's no other change that was near 20 along the graph, right? All right, so that's that for that. Now for the next part 3, is to suggest 2 reasons for the shape of the graph during each of the following time periods. Again, you can make a record of the graph itself, and then they come back to this section. And so from 6 a.m. to 8 a.m., I kind of indicate the change in mass right here. And again, you can go back to your graph to kind of confirm this as well. And so what you have here is a small loss in mass, which is 4 grams for each section, right? And so since the mass is so low in terms of change or loss in mass, then the reason for this may be because the temperature may be low. And since you have low temperature, then you have little evaporation. You have high humidity, and which means the air is moist. And if the air around the plant is moist, then you have little to no loss in water due to evaporation, all right? Because the atmosphere will be already saturated with water. Low wind speed. And since you have low wind speed, you have water will move slowly away from the plant. All right. And if you have low light intensity, then the stomata will not fully open, because remember that light stimulates the opening of the stomata, right? And so once the stomata, once they are closed, then you have a reduction in transpiration. Now between 9 and 11, which you have the highest loss in mass, and this again is 40 grams average between the difference in time. And so if this is the highest change in mass or loss in mass, then is due to high temperatures, again rapid evaporation, high wind speed, which means the release water or the water coming out of the plant or the evaporated water will then taken away rapidly from the plant, and so therefore it will increase the rate of transpiration. You can also have high light intensity, in that the stomata will be fully open, and then that makes evaporation much easier. A low humidity as well means that the air is dry, and since the air is dry, then you can have more water releasing into the atmosphere. All right, and at noon to about 2 p.m., pretty much the graph was maintaining at 40 grams. So it maintains the 40 grams, which means there pretty much no change, little to no change at all. No change, in fact, between that period, no change. So there's still 40 going straight across. Again, you can go back to the graph to confirm that. Now, the reason for this, it could be very low temperatures, not just low temperature, but very low temperatures. That means you have no evaporation taking place. You could have extremely high temperature as well, which means the stomata was closed. Because again, when the temperature is really high, the stomata will close to conserve water. Also, if the plant reach a wilting stage, that means no further loss in water or no further evaporation can take place. That means all the water in the plant already evaporated. All right, that means no other possibility of evaporation at all. The stomata could be closed as well, which means water is now be conserved. And this again ties in with number 2. All right, you can have rain in the atmosphere, which means that the air is moist. And so once the air is moist during rain, then there's no evaporation of water from the plant going into the atmosphere. You can also have still wind. So if the wind is not blowing at all, then that will cause the water to not move away from the plant. And so water stays on or around the leaf or the leaves of the plant, and hence no evaporation. So there are a number of reasons you can put on this or these sections, but again, they must make sound sense. They must be solid in terms of scientific knowledge or concepts. Now for part C, and part C1 is to discuss how transpiration helps plants to move water from their roots to their leaves. And so there are a number of events that actually take place here to make water move from the roots into the leaves. And the point to note is that water will come from the root hair cell, right? And remember that the root hair cell have a really large vacuole to kind of store water. And so the first thing may happen here, well, this not happen, there's a description of water, that water is really cohesive and adhesive. And so the principle of movement of water is based upon these two things. Now, one is that the warmer water will evaporate from the leaves, right? And so as water evaporates, the evaporating particles will pull other water molecules along, and this is due to water being cohesive, which means there are water molecules attached to other water molecules. And so if you evaporate one, as it moves away from the plant, it keeps pulling the other water molecules behind. And so once that's taking place, what we're going to have as a result is a continuous pulling of water molecules, and that is achieved due to evaporation, known as transpiration pull. So as evaporation taking place, you have a continuous stream of water known as transpiration pull. And now transpiration pull also aids in what they call capillary action. And capillary action occurs because of the narrow xylem vessels. And again, water is adhesive, so it attaches the side walls of the xylem vessel. And since the xylem vessel is narrow, then you have what they call capillary action taking place. And so water climb along the xylem vessel and also make it easier for evaporation and the continuous flow by transpiration pull to occur. So once you explain anything along that line, you should be good. All right, so there are a number of things I must mention. Movement of water, how the water is able to move, and through with structure and why. All right, for part 2, is that while absorbing water from the soil, plants absorb mineral salts such as magnesium, calcium, and nitrogen. Is a state one role of magnesium and one role of calcium in plants. And so for magnesium, it's really the production of chlorophyll. That's the main function. Yes, magnesium will help with other functions as well, but the main thing is to produce chlorophyll. Now, calcium helps to form cell walls, all right? And nitrogen is used to make protein here. There are just 2 reasons why plants need protein. So why plants need protein is for a number of reasons, and it is similar in most living organisms. And so here, plants need proteins to make enzymes, hormones to produce new cells for growth and development, and also for to make the plant generally healthy, and also in terms of immunity to fight against certain diseases or attack by certain diseases. All right, so enzymes, hormones would be the common answers that we'll expect from you, and also to produce new cells for growth and development. All right, but as well help with health and immunity of the plant. Now for question number 2, figure 3 is a diagram of the carbon cycle, right? And I kind of label some of these processes already. So when you go through, it's going to be easy. You have combustion, you have photosynthesis, you have respiration, you have decomposition. All right, and here's the name of the processes numbered 1 to 4. Again, you can go back to the chart and you will see them. Photosynthesis, respiration, combustion, decomposition, right? And for 4 marks. And then for part B, it's a name one type of organism responsible for decomposing organic waste. And we have, these are 3 possibilities here: bacteria, fungi, and earthworm. All right. And of course, bacteria will be our most common answer. That will be the common answer for most students. All right, for part 2 of this question, is explain 2 ways in which the type of nutrition carried out by the organism named in B1 is different from that carried out by plants. And so we're talking about decomposers. So whether or not you list the bacteria, the fungi, or the earthworms, then it'll pretty much be the same. And so the main thing here is that for the decomposers, whichever one you choose versus plants, no light is required for the decomposer. Light is needed to produce food for the plants. And again, for decomposers, it depends on other organisms. Even though they are depending on the dead portion or the dead organic materials, they are still depending on other organisms, while plants will make their own food. Now, in terms of decomposers, now they break down the organic molecules or the complex organic molecules into simpler molecules, while plants now, they use simple molecules to make organic molecules. And a point to note that the simple molecules that plants use will be the inorganic molecules, so they're changing from inorganic to organic. And the opposite is true for decomposers. They convert organic materials into inorganic materials. And so let me give an example here. So for example, decomposers will give you nitrates from proteins. So if proteins are broken down, they may get nitrates from the proteins. All right. And in plants, they may convert things like carbon dioxide and water into glucose, which glucose is an organic material or chemical. All right, let's go to the next part of the question now. And again, please observe the marks to kind of guide you in answering the questions. I cannot overemphasize that. Now, for part 3, it says state 2 reasons why the type of organism named in B1 is important in an ecosystem. And we're talking about decomposers again. And so decomposers will help to reduce the buildup of waste. Imagine without any decomposers, then dead animals, dead organisms, whether plant or animals, will just keep piling up. So help to reduce the piling up or build up of waste materials. Also help to recycle nutrients. All right, and so nutrients are made available to producers. So when bacteria or mushroom or fungi generally, or even earthworms, once they break down these dead materials, they now convert into richer soil. Example, making humus, and humus is rich in nutrients that is good for other plants or plants generally, which are producers in food chain. And once you have good producers or high energy producers, then you have more viable food chains and food webs, or you have more energy available to organisms within food chain. So that can be explained in so great depth. And then next one is to reduce the spread of diseases, definitely. Because if you have dead materials or dead organisms lying around, then the spread of diseases will be much greater. All right, and also aids in the flow of energy in the ecosystem as well, which ties in pretty much with number 2. All right, so again, you're only looking for 2 marks, and it said state 2 reasons. So you just go through and remember the best possible one. You may never know the exam may ask you for 3 next time. All right, so just 2 reasons why carbon dioxide is described as a greenhouse gas. And so I have 3 points here. One is that carbon dioxide is able to trap heat in the atmosphere. So it helps, carbon dioxide pretty much warms the earth, and that's a characteristic of a greenhouse gas. It also helps in terms of absorbs heat energy, which is radiated heat or infrared radiation. Carbon dioxide also radiates heat to the earth, which means it emits light. All right, so it not just only absorb, but it also emit light as well, or heat. All right, and so for those reasons, carbon dioxide is classified as a greenhouse gas. All right, so part 2 of this question, and I make sure I highlight this, is with reference to the carbon cycle. All right, so basing this on the carbon cycle. So just 2 strategies individuals can use to reduce the amount of carbon dioxide in the atmosphere. We're not saying that these are logical in terms of, we'll not even use the word logical, but practical. It only says based on what you see in the carbon cycle, suggest now 2 strategies that can be used to reduce the amount of carbon dioxide in the atmosphere. And so one is to plant more trees, because trees are what taking away carbon dioxide from the atmosphere. 2, reduce combustion. 3, reduce decomposition or reduce death. Again, just to make a note right here, the last one is to reduce respiration. And of course, if you reduce respiration, then the organism will lack of energy and will die. But based upon the carbon cycle, respiration is producing a lot of carbon in the atmosphere. So to reduce it, you have to reduce the possibility of respiration. Of course, that's not a viable way to go about life, right? That's why I even put that as number 5. All right, so a motherfucker, I could put this in red because this is not a possible thing, but based on the carbon cycle, yes. All right. All right. And again, just want to see if you can analyze a situation. So this is a question to think about, critical thinking technically, or just to look and analyze a particular situation. Now, for question number 3, is a figure 4 shows a diagram of the human respiratory system. It's a label on the diagram the 4 structures that are directly involved in breathing. And for you to breathe, then you need your nostril or your nasal cavity, because that's the main passage for air to enter your respiratory tract. You need your trachea, and you also need a bronchus or bronchi for both of them, but you only see one here, so it's bronchus. And you need your lungs as well. All right, so those are very important for breathing. All right, you may say you can breathe through your mouth, yes, but you don't naturally breathe too much through your mouth, because if you try to close the nostril and breathe through your mouth, you're going to realize that breathing become really uncomfortable. All right, or your body starts to strain. All right, but you can do breathe through your mouth as well. All right, but it's not a main part. Now for part B, is a state how any 3 of the structures labeled in A function in breathing during inhalation. So in inhaling, the nasal cavity will intake air, and what the nasal cavity will do is to filter the air, moisten the air, and also warms the air. All right, the trachea now will direct inhale air towards the lungs, or you can direct air towards the lungs, and it also further moisten and warms the air as well. That's the purpose of a trachea. They are the function of a trachea. And the bronchi, they passage the air from the trachea into the lungs, all right, or into each lung. And the lung now will collect the air, which is oxygen particularly, and absorb oxygen and make it available for the body. And of course, you know that you'll have gaseous exchange taking place in the alveoli. So the alveoli is not for breathing, but the alveoli, they are for gaseous exchange. All right. Now let's go to the next part of the question. Part C is to explain how any 2 features of the human lung make it an efficient respiratory surface. And to make it be efficient in respiratory, as a respiratory surface, is one, it must have a large surface area. All right, and that's why your lungs are really large in the body. And so large surface area increase the volume of oxygen that can be absorbed, taken in and absorbed. Moist, and to be moist, this increases the absorption property or the absorbing property of the lungs. And many folds, so the many folds inside the lungs, you have a lot of alveoli as well, which we're going to talk about next. So it could have many folds in the lungs, you know, and that will increase the surface area and also an effective absorption property. And many bronchioles and many alveoli as well are very important, because the many bronchioles will help to distribute air rapidly throughout the lungs. But the bronchioles are doing that. And then the alveoli, the many alveoli will increase the effectiveness in gaseous exchange. All right, so you could separate bronchioles and also alveoli, because again, as I said, bronchioles will kind of distribute the air throughout the lungs, while the alveoli will increase the effectiveness in gaseous exchange. Many capillaries is also important as well, because the many capillaries will also increase the rate of gaseous exchange. Because remember, the capillaries is important in carrying the deoxygenated blood to the lungs and carrying back oxygenated blood back to the heart. So the more capillaries are there, is the greater in terms of effectiveness in the gaseous exchange. Now for part D, it's a breathing rate can be affected by several factors. Explain how exercise and smoking may affect a person's breathing rate. And so effect of exercise, and what the exercise generally do is really to increase the rate of breathing. And so during exercise, the body works harder and require more oxygen, definitely. So hence breathing increase the supply, the extra oxygen needed to make the required energy. All right, because more exercise means you need more energy, and so therefore more breathing for the purpose of supplying more oxygen. All right, and so increased respiration will equal to increased oxygen requirement, and so therefore you breathe to compensate for that needed oxygen. All right, effect of smoking, which is a bad thing. This could lead to serious problems, health issues such as cancer, and so on, emphysema. Now, smoking causes the buildup of tar in the alveoli. That's one major thing. And the buildup of tar reduces gaseous exchange, resulting in deprivation of oxygen in the body. And so breathing will increase to compensate for this reduction of oxygen diffusing across the alveoli into the blood. Now, point to note is that, however, if smoking caused structural damage, which means the tar built up over a period of time and start to damage the structure of the alveoli or the lungs, then what will happen here? It results in shortness of breath. All right? And the reason why you end up with shortness of breath because you're unable to keep up with oxygen requirement, in that oxygen cannot pass over into the blood that easily or effectively. You have a lot of carbon dioxide in the blood staying in the blood just the same, and so you realize that the body will become deprived and struggling to send air or oxygen across from the alveoli into the bloodstream. All right, and so even your brain become affected by that because the brain have less oxygen, and then you can also faint or pass out. Now for section B, question number 4 is to describe 2 events that occur in each of the following stages of meiosis 1, and that is meiosis 1, just to make a note. And so for prophase 1, and remember that is PMAT, all right, and you have IPMAT, which is interphase first. You only have one interphase in meiosis, remember that, all right? And so in prophase 1, the chromatins, keyword here, the chromatins will condense into what they call chromosomes, all right, because the chromosomes now become visible in prophase 1. The nuclear membrane start to disintegrate, very important. Homologous chromosomes come together, form what they call tetrads, and while tetrads are forming is a process called synapsis. All right, so homologous chromosomes exchange genes, which we call crossing over. All right, so the coming together of the chromosomes, the homologous chromosomes, we call that synapsis, and a tetrad is formed. And then now, when homologous chromosome exchange genes, that is what they call crossing over. So know the difference between synapsis and crossing over. Now the centrioles will move to the opposite side or opposite poles of a cell, and then you have spindle fiber will start to form. All right, and of course the spindle fiber will connect from the centriole all the way to the centromere of the chromosome. All right, for the anaphase now, the tetrads will separate. All right, and at this point you have exchange of genes already taking place. The spindle fiber will become shorter, and because of the shortness of the spindle fiber, then the homologous chromosomes will move to opposite poles. And then point to note here is that sister chromatids remain attached to their centriole. So you're not going to have chromatids moving to the different poles, as in mitosis, 2 different things, all right? All right, and so in the telophase 1 is that the nuclear membrane reforms around each set of chromosomes. Notice the keyword here, chromosomes, not chromatids. Now the cleavage furrow will also form, and cytokinesis will begin. Once cleavage furrow start forming, then we have what we call cytokinesis start to form. And this is when the cytoplasm start to divide. And then we have the nucleoli start to reappear. All right, and that's 6 marks. So 2 marks, 2 points for each thing as they suggest in the question will be fine. All right, for this 6 mark. Now for part B and part B1, it said mutation and sexual reproduction are 2 processes which can lead to variation in the population of species. State 1 way in which both processes are similar and 2 ways in which they differ. And so in terms of similarity, one basic similarity, are pretty much the only similarity between mutation and sexual reproduction is that they are random processes. All right, they're really random. All right, and so let's talk about the differences now. And so for mutation, the DNA sequence changes. For sexual reproduction, the DNA sequence remain unchanged. For mutation, chromosome number or form of chromosome may change. However, no change in chromosome number for sexual reproduction, because what you're going to get as a result is the same combined number, even though the only part in sexual reproduction, we have a change in chromosome number is when they actually form the gametes. But once gametes are formed with the proper half or applied, then they come back together, give you the full number, which is the diploid. So technically, there is no change in chromosome number. Now, for part 2, it's to explain the difference between variation in a community and variation in a population and suggest one way in which diversity benefits the community. And so let's talk about the difference now in community versus population. And just to make a note that a community is a group of populations, all right? And so now what is important for community to understand these differences is that in a community, you have a difference in species. However, in a population, you're dealing with the same species. And this bit of information is just to help to guide us in terms of the differences. And so one thing is that the size of organism differs greatly. And so if you have the same population, the size differs to a smaller degree compared to community. Because guess what? For example, you have a community with rats and dogs and elephants. They're much bigger and different. But if you have a population of only dogs, how much dog can be bigger than the next dog? Of course, some dogs are really bigger than each other, but generally, you have slight changes in size compared to community. Generally, no, you have difference in number of chromosomes because each organism or each species will have their own specific set of chromosomes. However, if the same population, if it is a population, which means the same species, that means they have the same number of chromosomes, right? And the feeding patterns will vary in a community because different organisms will have their own niche, which means they have their specific role. And so for that reason, they will eat different things. All right. And of course, for the population now, you have similar feeding patterns. All right. Again, this ties in with number 3, different niches. And we have the same niche for population, right? If you're a producer, you're a producer. If you're a primary consumer, you're a primary consumer. But of course, for community, they will be at different positions within the food chain or food web. All right. And now for number 5, different physical structures for a community, but for population, the physical features will be similar. And so let's take, for example, we have different in terms of number of legs, but however, for population, it must have the same number of legs. Also, a different number of petals, for example, flowers. And so if it's the same flower, which is the same species, then the number of petals must be the same, right? So if you compare dicotyledon versus monocotyledon, then yes, the petal numbers or the arrangement of petals will be different. All right. And also the art structures, for example, animals, they have different art structures. But if it's the same species, the art structure will be the same. So these are just some examples of different in physical structures. So just to be specific, you do not want to just say different in physical structures. That kind of vague and too open. So you want to narrow it down to a few examples, right? Now, a very important point to note here is that the variation in a community is wider compared to the variation in a population. So you can even start out with that in terms of explanation and then just narrow down to the list I gave you up top. And of course, you're not limited to only the things I gave you. Of course, definitely there are more options to this. Now, benefit of diversity to a community is one, if the more diverse a community is, it reduces the possibility of extinction. Why? Because you have a greater stability, and the greater stability is based upon a wider range of food sources, right? Because the more diverse it is, then you have more interaction between organisms, and so therefore you have more possibility of food sources. You have also greater adaptation as well and recovery from environmental changes, because the more organism you have within a area, and if you have any form of environmental changes, they tend to adapt better because they're so related or so closely related to each other. Then they kind of overcome the same environmental trends or changes. All right. So those are very important points in terms of benefit of biodiversity. Part C, and it's a briefly explain how bacteria may become resistant to antibiotics. And so just to run through this, and so we have here two ways. We have mutation or natural selection. So there are two possible ways that this can happen. So just to make a note, so it can either be by mutation or natural selection. So whichever one you choose to explain, that is also fine. Now, in terms of mutation, what will happen is that mutation occurred. And because mutation occurred, you have resistant bacteria forming. And so introduction of the antibiotics now. And so the unmutated bacteria, they are generally killed because the antibiotics will be specifically designed for those bacteria. And so the mutated bacteria will survive and divide by cell division, of course, binary fission if it's bacteria. And then the antibiotics now will have no effect on the mutated colony of bacteria. So there now you have a resistant strain bacteria. In terms of natural selection, the very first thing to note is that you have variation in bacteria colony, which means you have different types of bacteria. So some bacteria are resistant, while some may not be. All right, and so introduction of the antibiotics, the resistant one will survive and they will reproduce by division, and then the resistant bacteria will divide again and continue with the colony and so on. All right, so any of the two ways, then you will be fine. All right, and we are jumping into our second to last question. And this one to describe the location and structure of the normal human female reproductive system. And the human female reproductive system is located in the pelvis cavity. That's the location. And so we have a number of structures to talk about here. We have the labia. And the labia is a protective skin that protects the opening of the vagina. And that is the outermost part of the female's reproductive system. We have the vaginal canal, or what we call the vagina, as a tube-like structure that leads from the outside to the cervix, and it receives the penis as well, and also receives semen. The cervix now, which is also called the neck of the womb, it is located at the end of the vagina and the base of the uterus. All right, and so the uterus now is a hollow pear-shaped organ that accommodates the development of the fetus. And we have the fallopian tubes. And the fallopian tubes, they are like structures that connect, they are tube-like structures. So it is supposed to be tube-like structures here. I must put that in. So they are tube-like structures that connect the ovaries to the uterus. And then we have the ovaries now. And the ovaries, they are ovally shaped. So the ovaries are ovally shaped. That's a nice way to look at it. Ovaries are ovally shaped. Yep. And they secrete hormones and also secrete egg as well, or the production of eggs. All right. So let's now slide down into part B. And for part B, it's a newly married couple want to delay having children for a few years. Suggest one temporary contraceptive method that the couple could use to prevent pregnancy at each of the following stages. And so to prevent fertilization, condoms, definitely. You can use cervical caps. You can use diaphragm, or you can do the withdrawal method, or better yet, abstain. All right, but if they're a couple, then that may lead to problems as well. Now, the next one is to prevent ovulation. To prevent preventing ovulation, you use generally chemical means. So this will be oral contraceptives or injections. So you can use the pills. That is the most common method, using pills. So anything that are hormonal based, pretty much, that will affect the release of egg during the menstrual cycle. Prevent implantation, IUD. Even though IUD can use for other purposes as well, but for implantation, IUD is most effective out of all for that purpose, preventing implantation. All right, the egg reaching the uterine wall. For part C, explain how each contraceptive method suggested in B works to prevent pregnancy at each of the following stages. And so preventing fertilization, what they do, any of the one that you list, they prevent the sperm cell from reaching the egg. That's basically what it's doing. So example, a condom will captures and collect sperm cells or semen, preventing them from fusing with the egg. All right, so that's how condoms, for example, will work. Caps will do the same thing. They prevent the sperm cells from reaching to the egg cell in the fallopian tube. Now, preventing ovulation. Pills are generally called hormonal contraceptives. They alter the progesterone level. And in fact, what they do is they increase the progesterone level. And so higher progesterone levels now cause the uterus to become more thicker. All right, and this also prevents the ovaries from releasing an egg. So just like when a female is pregnant, it's going to mimic a pregnancy in a sense, because if a female is pregnant, then she will not ovulate. All right, and so it's kind of the same effect with the high increase of progesterone. It kind of tells the body, hey, listen, you're technically pregnant, right? So the walls are thick, so the walls are going to break down at this point in time. So it kind of mimic pregnancy. Now, preventing implantation, again, how this works is that the IUD prevents the egg from implanting in the walls of uterus. And what IUD does is to kind of block the egg, right? Or it also changes the structure of the uterine wall as well and preventing implantation. So IUD help also to build up the secretion of mucus, and that will also cause blockage of the egg onto the uterine wall. All right, so we're on our last question now. And this last question is to state the name of the final products of digestion for each of the following types of nutrients. And we have carbohydrates, and we know that the end product for digestion of carbohydrates is glucose. And the word they could also use right here too is the hydrolysis of these things instead of using digestion of. All right, so proteins, we will get amino acids from protein. Fats or lipids, we'll get our fatty acids and glycerol. All right, and that's just a 3 mark right there. All right, for part B, it says in addition to the macronutrients in A, list 3 other types of nutrients in a diet that would promote the maintenance of good health. And so here we have vitamins, we have minerals, we have water, and fiber is also one. But the reason why I put fiber in red, because fiber is also a carbohydrate. It's a very complex carbohydrate. And guess what? We cannot digest fiber. It is cellulose, it's roughage. So we are unable to digest cellulose, all right? But indeed, it is very important for maintenance of good health. Prevents constipation, allow the easier digestion of food, allow food substances to move easier along the alimentary canal, all right? And that promotes healthy digestive tract, all right? So even though we can't digest it, but it's very important to maintain good health. All right, and so we are on part C. It says Mary is diabetic, but her husband Marvin is not. Explain how the concentration of sugar in Marvin's blood is kept within the normal range several hours before and after he eats a carbohydrate meal. And so before he eats the meal, here, what happened here is that the stored carbohydrates in the form of glycogen is available when glucose is low in the blood. So before you eat that meal, it may have low glucose in the blood. So what will happen here is that when glucose is low, the pancreas secretes glucagon. And glucagon converts the stored glycogen to glucose. And so the blood sugar will increase to normal. That's very important. So you have an increase in blood sugar, but it increased to the normal point. However, now after carbohydrate meal, what will happen here is that the excess carbohydrates, particularly glucose, is sent to the liver, and then insulin now is released from the pancreas, and insulin converts glucose into glycogen, and glycogen is now stored in liver, and this decreases the glucose to a normal level, okay, or decrease the glucose level to normal. All right, and that's our 6 mark question, and we on the next section here. Going on, well, this is the last part. It's to explain why Mary is advised to limit her consumption of simple carbohydrates. And simple carbohydrates, we're talking about her monosaccharides or glucose particularly. So she's unable to effectively convert glucose to glycogen because, again, simple carbohydrates, they are easily absorbed into the blood, all right, are easily broken down and then absorbed. And so hence consuming too much simple sugars or simple carbohydrates will increase her blood glucose level above the normal. And again, if she's diabetic, she's going to be very difficult to convert the glucose into glycogen, right? So she's unable to do so. And so this will result in many biological conditions or complications. So you don't want her to have many medical problems, such as feeling fatigue, drained, tired, sleepy. If she get a cut. So there are many different effects of diabetes you can search on. So please read on diabetes. know the effects of diabetes and the symptoms of diabetes. And at this point, I want to thank you for watching this review. And please, if you haven't done so, subscribe and share with your friends as well. And I wish you good luck on the examination. Take care, and we will talk soon. All right, have a great one.